So in these videos, when I try to talk about hard topics to explain, like most of them are in science, like for example, the last one I tried talking about in chemistry, I tried to make it a little bit easier for some people to understand. Try using different analogies or just trying to very much just re-explain, maybe just simplify how I'm going to do it. So, so today I'm going to talk about viruses, like I said, but viruses is such a broad and big spectrum to talk about. So I believe this will be like part one of maybe a 2 or 3 part video, 2 or 3 part like videos. So so this won't be entirely everything, but this will get you a very good start in what I'm trying to get at, basically. So let's begin. So obviously, some of the things you might be curious about is what exactly are viruses? Now, viruses are much smaller than cells, and they aren't actually cells. And so viruses, just like cells, come in all different shapes and sizes. Some of them I'm sure you've seen maybe like this. Maybe they look like that. Maybe they just look something like that. Or maybe they look something more like this. You know, these are all correct forms of viruses, but these are actually different kinds of viruses. For example, this one might be a bacteriophage. This one can be any other virus that has spikes and an envelope, which I'll get to. And this one it could be just the same thing, another thing with an envelope, but then it's also showing the enclosed parts of it, such as capsids or maybe the nucleic acid inside. So these are all appropriate forms of said viruses. Now these viruses are sort of kind of like, you can kind of relate them to something like a parasite. And basically they infect like a host cell, or they infect most of the time they infect a host cell, which usually affects the actual host or being. So sometimes you can get sick with viruses. Like for example, the flu is a virus, or maybe something more dangerous like Ebola is also a virus. So these are certain things that you should consider. Okay, so for example, basically trying to explain the composition. So basically, most viruses are enclosed in this big envelope, and within the envelope we have this thing called the capsids, which are actually just a bunch of things called capsomeres. But inside that we have our nucleic acid. So here I'll just draw it like that, I guess. So let's say that is the nucleic acid. Inside here is the nucleic acid. And here, where I'm drawing all the circles, are something called capsomeres. And the entire thing is called capsid, usually. So capsids. And so basically these capsids are what encloses the nucleic acid. I'm sure you know what nucleic acid is. Basically DNA or RNA, and it's used to put inside host cells, such as like a normal cell, and go into that nucleic acid and replace or sometimes just permanently or temporarily, depending on the virus, its nucleic acid with the cells, and therefore the cell can make more of these, and that's how they multiply. Basically, these viruses go into cells and they replace their nucleic acid, and nucleic acid is basically DNA, or deoxyribose nucleic acid, or RNA ribose nucleic acid. So that's what the NA stands for in DNA and RNA, nucleic acid. So that's why we just say nucleic acid. Now, one thing to consider about viruses is that they only have DNA or RNA. They never have both. They only have one or the other. And DNA usually looks like that, I guess. Yeah. So that's usually how DNA looks, like a double helix, and RNA is usually half of that. It usually looks something like that. And obviously there's like the guys like that, like kind of like a twisting ladder, and then the RNA just looks like, like that, kind of, as if it's like half of it. You like took half of it, and that's what you got from it. Just basically one of the strings, and that's RNA. So yeah, and so that's the capside. The capsides are usually an important part because capsides are usually what determines the shape of the virus. And it basically encloses a nucleic acid, because you can't just have nucleic acid floating around. So yeah, and then here is the envelope. Now, not all viruses have envelopes. Now, there are certain differences between these. So basically, most envelopes are acquired after they're already like put through the a cell, and basically the envelope is usually a part of the cell membrane. So for anyone who's been in like 6th or 7th grade, so here's like a cell, here's like a rough thing of a cell, maybe that's like the nucleus, but it has like a cell membrane, which is basically the cell walls. And so basically after the virus gets out, it's going to obtain part of the cell wall, and that's going to be the quote-unquote envelope. So yeah, and there are certain advantages and disadvantages of having this envelope. For example, some of the advantages that would be pretty nice. For example, this makes the, let's say the host figures out that, the host cells figure out that there is an intruder, the virus. And so when they start looking for it, and some of the viruses may or may not have the envelope, the ones that do not have envelopes will start getting attacked, but the ones with the envelope may not be attacked, and they can be hidden from the attack, and they can sort of like hide away and not get hurt. So that's one of the advantages. But another disadvantage is that they can get damaged more easily. So for example, outside effects such as temperature, whether it's too hot or too cold, or maybe the pH level is different, or maybe the chemical composition around it. It's really just outside effects that could really infect the envelope. But ones without the envelope are totally, these effects don't matter to the ones without envelopes, and they're totally fine. And last but not least, some of them also have these little things called spikes. Now, the spikes are kind of useful. It really depends. Now, most of the time, spikes can be, they can actually, again, just like the envelope, can be worked both ways. Like, for example, the spikes, they can, if they are touching maybe some of the cells, if they touch some of the cells, they can identify themselves between which cell, because sometimes not all viruses can go to specific cells. I'll go through that just a little bit later. But basically, it is really good to help identify which kind of cell it's trying to attack, or which one it can attack. So that's good for them in that case. They can help identify without trying to constantly go through one single cell, even though it's not compatible to go inside with it. Or, for example, whenever there are red blood cells passing by the virus spikes can touch if they touch the red blood cells, most of the time they can cause clumping, and clumping of these red blood cells, and the body can look at this, and it's another sign to see, oh, there's an intruder inside here, right? So basically, spikes and envelopes, they don't always occur in viruses, but capsides, I spelled that wrong. Capsides or nucleic acid can go in between which one? Either one. Okay. So yeah, next we're going to talk about, and shapes and sizes doesn't necessarily, it's not intensely important, because all you have to know is that there are different kinds, or different shapes and sizes, depending on the amount of capsides and capsomeres that there are, like I was talking about earlier. But now let's talk about host range. and specificity of viruses. Specificity of viruses. So basically host range is referring to what kind of host can virus go. So for example, if you know about Ebola, and I'm not sure if I talked about this or not, I believe I have in a very old video, you can go check that out. I did talk about viruses. And basically host range is what exactly can the virus attack. So basically, for example, some viruses might be able to jump from a monkey to like a human, maybe back to a pig, like for example Ebola. But like there is a specific chimpanzee virus, one of them that is called, for example, polio viruses. They can be grown in like monkey kidneys, but not exactly, but it's not like really affecting any of the people who are like researching them. So they don't really need to put a bunch of protection to not get, viruses inside them from all the research, right? So that's basically host range, and specificity of viruses. Hopefully by the next video I can try pronouncing that correctly. Specificity of viruses. Basically, what kind of cells can the virus hurt? So for example, you might be able to hurt tissue cells, or maybe some muscle cells, or it could be like red blood cells. There are different kinds of cells, and some viruses, just like Ebola, can attack all of the cells, but there are other ones that only attack specific ones. Some of them only being able to attack like tissues, or some other being able to attack only organs. So again, host range and specificity of viruses, those are two separate things. The difference is host, you just need to know that it's the kind of thing, animal, creature. Sometimes it can even hurt like plants, or some measly tiny thing like bacteria. The host range is what can a certain virus attack, what kind of person or thing. And specificity of viruses is talking about what kind of cell. So can it take like just a normal cell, the ones like for maybe perhaps for like the muscle, or maybe for the skin, like maybe a tissue one, or maybe red blood cells, maybe the ones that are actually for the blood. Those are the kind of things that you gotta look out for these two, because they're very similar things, but they talk about two different things, to be specific. So yeah. And last but not least, we'll talk about classification of viruses. So basically, by looking at the name, you can tell it depends like what classifies each virus, because before most of the virologists were able to know the structure or the chemical properties of such viruses, they basically classified them on what kind of host that they affected, or perhaps maybe the structures that they affected. So the viruses are classified by bacterial, as bacterial viruses, maybe also called bacteriophages, like I said before, the little guys that kind of look like that. Plant viruses, so basically they attack plants, or animal viruses, which they attack animals. So basically in these three categories. And animal viruses are a little bit special because, again, with animals, they have so many more kinds of cells. For example, they have skin cells, so like tissue. So if they attack tissue, they were called let's spell that right, germotropic. They can attack if they hurt skin, or if they did tissue, but skin itself, which is a little bit different, is neurotropic. I guess neuro, like brain tissue, perhaps. Or if they attacked nerve tissue, so that would be a viscerotropic. Or if they attacked oh wait, so sorry, this one is for tissues. Yeah, so this one is for tissues. This one's for skin. Or no, this one's for if they attack nerve tissues. That one's for skin. So this one's skin. Skin. Sorry, I keep mixing them up. So that one's skin. This one is for the nerves. This next one is basically organs, or the digestive tract. So organs. And last but not least, oh, you almost couldn't see it. If they attack respiratory, it would be pneumotropic. I believe the P is silent. Pneumotropic. If it's the respiratory. Or the lungs, basically. So, most likely these pneumotropic, if they attack the respiratory, most likely they are airborne. Basically, you breathe them in. Maybe they come from different kind of plants. Maybe they come from, most of the time they come from a different species. Or they, if they're only single species, they start attacking you. So maybe they're from another person, maybe perhaps like the flu, for example. So yeah, this is basically all I have to talk about for viruses right now, because there's such a right now in a bio book that I'm trying to read right now. So here, this one's about microbiology. I'm trying to read the chapter on viruses right now, classification of viruses, and so this is a really big chapter to like kind of go through, and I didn't see any sense of trying to make it right now, and as you can see, I'm already getting pretty close to like 20 minutes about. So yeah, that's basically it for my videos. If you like this, please do look. I do try to do other videos like this, and I hope to see you guys later. Goodbye.